and limited in vitro expansion capability. So these are the cells. Now the question is, do lungs have stem cells? Any idea? Yes, lungs do have stem cells, and you can see this is the distal lung, and you know the alveolar spaces, which is important for gaseous oxygen diffusion, and this is the proximal stem cell. I have been working mostly on the proximal stem cell, proximal lung, and it is called trachea. You can see the section here drawn, morphological features of that, then the small airways, and then the alveolar structures. If you have to do cross sections of this, you can see how they look like in the cross section. So this is the epithelium, pseudostratified epithelium, and you can see at the base the stem cells are here. The yellow colored cells are the stem cells. And in this also we can see some basal cells, but the basal cells don't exist in the alveolar space. And there are other stem cells there. But as my work is in the area of this, I will be concentrating more on this adult basal stem cell. So why lung stem cells are important in research? As you can see, this is the uninjured epithelium. This uninjured epithelium, when it gets injured, then the epithelium gets sloughed off, and the stem cells then divide over time and differentiates and finally reforms the epithelium. So it's normal injury repair. So how do we model this? We use a compound called polydopterin. Polydopterin is a surfactant when injected inside the trachea through intratracheal delivery. The polydopterin sloughs off this epithelium, and we can create a model of injury and repair. And we can see how the normal repair happens. If the normal repair goes aberrant, what happens is we can see there is a stratification of the K5 positive stem cell, and there is a loss of differentiated cells, and this is a hallmark of premalignancy. Now, if these stem cells, K5 positive stem cells, breaches the basement membrane, then it's a hallmark of malignancy. And stem cell differentiation, stem cell proliferation and differentiation defects are seen in all types of lung diseases. So that's why lung stem cell studies are very important. So we will show in the next slide that these basal stem cells are very important. These basal stem cells actually can lead to all cell types of the airway epithelium. So this is a confocal image, and you can see the basal stem cell in green at the bottom of the basement membrane, bottom of the pseudostratified epithelium sitting on the basement membrane, and these are the differentiated cells. So these cells are very important for this, and not only because they can produce all this, but they have other functions also. So how, I asked this question, how adult stem cell maintains the tissue homeostasis after injury using lung as a model system. So now we can see, we are interested to understand how lung stem cells are associated with lung cancer. Now, lung cancer, as you all know, is the deadliest form of cancer worldwide, and the ICMR forecast a 7-fold increase in the number of lung cancer by 2025 in India. So it is growing alarmingly, and you can see the morphology of this lung. And here the differentiated cell is there in normal airway epithelium, and these are the stromal cells, and you can see again the airway stem cells at the base. Now, as I said, if the stem cell proliferation increases or the loss of differentiation happens, these stem cells form a premalignant lesion. There is a cartoon here to explain this image. These are the confocal images of the respective sections. And when it reaches this, then they form tumors in the lung. So these airway stem cells can form squamous cell lung carcinoma, and this is a direct correlation to this. So they have a nexus with the bad man, and the bad man gives them feedback, and they don't do anything. So there is a nexus going on, which we are finding it out, and how it's exactly happening, we are trying to understand. So here we have now taken this human normal airway epithelium, premalignant epithelium, and squamous cell lung carcinoma, and now we stain with this specific beta-catenin, which is phosphorylated, and Y489. Now you can see in the normal epithelium they are not there. Y489 is not visible even. And during the premalignant cells, you can see many of them has nuclear Y489. It's very interesting. Now in the cancer also they are significantly high. So you can see their levels are changing. Beta-catenin Y489. So this is an interesting marker which can be used as a biomarker, or we can target We understood that. Now, also the porcupine expression in these cells also, and you see porcupine expression in these cells from the normal, it is increasing in the premalignant, is high in the tumor. So again, porcupine and the beta-catenin Y480 both are going in the same direction, suggesting that if we target porcupine or with beta-catenin, especially Y489, we can probably take care of lung cancer, squamous cell lung cancer carcinoma. So we modeled this in a dish, and as you can see here, that we have modeled this. We use CHI. CHI is a compound, chemical compound, which activates Wnt beta-catenin pathway, and we are taking this stem cell, growing them, and then we add the CHI, so we increase the proliferation. So this is the in vitro model we made using mouse and human airway stem cells to model this and check for drugs. So now we thought that if we can find a inhibitor of Y489, that will be probably a very interesting drug that can stop lung cancer. So here you can see this is the model, and we have once we activate the CHI, the Wnt beta-catenin pathway increases significantly. Now we did a high throughput screening, and we found a compound called WIC1, Wnt inhibitor compound 1, that actually is reducing the CHI effect. CHI is an artificial chemical which can activate this Wnt pathway, but this WIC1 at a very interesting 10 micromolar also, it reduces this whole hijacked by Wnt CHI, this reduces significantly. So this and a group of compound which we patented are very interesting, and this compound is now in the market. We didn't want to go for the clinical trial because it takes a long time. So we sold this compound in the market. Now you can use this compound, you can buy this compound and use it for understanding the Wnt beta-catenin pathway. So this compound is available now in Sigma, Topris, and all other compounds. And you can see our paper is mentioned here with this. And we also work on other 3D disease models, which I want to just show that we are working on organoid models. And organoid models can be isolated from the stem cells can be used, or tumor cells can be used to make these organoid models. Some of these organoid models we developed to model pulmonary diseases. You can see this. This is the trachea of a human. We chop it into pieces, we cut out the stem cells, we put them to make organoids, and you can see how they look like. They looks like almost like this, the normal epithelium. So we modeled these organoids. Very recently, we also made lung organoids for the first time using multiple cell types. These are iPSC-derived cells. We are also fetal lung fibroblast. We have used umbilical vein endothelial cells, small area vein epithelial cells, and other cell types to generate airway models like this. And now we can generate them in 96 well plate like this. And this went like a lot of appreciation we got to many. And now we have modeled this, use this to model different diseases like lung fibrosis and other diseases. We have published multiple papers in this domain, and we can help others also to develop this into lung models, and also this can be used for other models. So I really want to thank my team, and here I have now started collaborating with multiple people. I'm lucky that they at least feel that I can do something, and they are collaborating with us, and I'm thankful.